All right, let's take a look at the process of translation itself. Okay. Now we said it's a 3-act play, so let's look at Act 1 first. Initiation. Oh, one last thing I do want to mention about ribosomes. These kind of experiments where you're taking ribosomes apart piece by piece, resealing them. One thing that was found a long time back is that you could remove 95% of the ribosomal proteins, and that would really screw up their function a lot. One thing that was not affected was the formation of peptide bonds, the covalent attachment of one amino acid to another. So people started thinking, is it possible that what catalyzes peptide bond formations is an RNA molecule, not a protein? In other words, is the ribosome a ribozyme, a catalytic RNA that makes them one of the most important processes in life, assembles proteins together? When they got the quaternary structure of prokaryotic ribosomes about 5 or 6 years ago, the answer was a definitive yes. They know exactly where on the ribosome peptide bonds are being formed, and there is no protein within about 2 nanometers of the place. So there's no protein anywhere close enough to catalyze that. So it's got to be that RNA. The other thing is with that 28 and 23S in prokaryotes. There are 2 sequences. There are a few tens of nucleotides apiece, and they're separate from each other. And now you're talking, these RNAs are about 2,000 to 3,000 nucleotides long. They're quite large, okay? But there's a few tens of nucleotides in 2 separate sequences, and those 2 sequences are absolutely invariant in every organism they look at. Not one single nucleotide change in those 2 areas. Now the rest of the molecule is pretty divergent. That's why you can use ribosomal RNA sequencing to do phylogenetic trees and things like that. Okay, but those 2 sequences are absolutely unchanged. With genetic engineering tricks, people tried changing one single nucleotide, and either of those 2 sequences completely abolished peptide bond formation. They killed the ribosome. So what that means is those 2 sequences must fold up in that big ribosome RNA and form the active site that catalyzes peptide bond formation. And it's so fine-tuned that any single change in it immediately kills it off. So that's pretty interesting. So the idea of catalytic RNAs, this is one of the most fundamental properties of life, the ability to make proteins. So this catalytic RNA are probably a much simpler precursor that could somehow catalyze amino acid formation to make peptides and small proteins. That goes back to the dawn of life on stuff, perhaps the boundary between living and non-living. And you're talking 3.8 billion years ago, possibly even a little further back than that. We don't have that in the fossil record. But, so this is a very fundamental thing. To this day, we still use a very important ribozyme to do one of the most fundamental properties of life itself, the synthesis of proteins. Okay, well, let's look at initiation, folks. Okay, the first thing we have, here's our messenger RNA with our cap and our coding sequence, and here's our AUG, and okay, here's the messenger RNA. First thing that's going to happen is an initiation factor that is commonly called a cap binding protein, or CBP. It's also called EI, eukaryotic initiation factor, EIF something or another. But it's an initiation factor. First thing that happens, the common name like is a cap binding protein.
Dr. Sauterer.
Yeah.
What is the small letters above the...
That? AUG, initiation codon. Codes for methionine. Now, by the way, in prokaryotes, it codes for a modified version of methionine called informal methionine. That's another difference between prokaryotes and eukaryotes. Same initiation codon, different amino acid, or different version. Okay, so our cap binding protein, it specifically recognizes that 7-methylguanosine cap. That guanosine cap has to be methylated, will not bind to an ordinary guanosine cap, and we can see that that will be used to regulate translation in some cases. Have an incomplete cap, the cap binding protein can't recognize it. It will not recognize other caps like the trimethylguanosine caps that we see in the small nuclear RNAs. And of course, if there's no specific cap at all, it don't know it's there. So that's the very first step, our cap-binding protein associates with it. Now, that carries it to the small subunit of the ribosome. And the small subunit is going to bind. Okay, now what I'm going to do here, I'm going to erase the messenger RNA looking like this, and we're going to have a little portion of it. Alright, and now we have I'm putting a few codons down there. Okay, the small subunit binds, and the small subunit then kind of walks down, or crawls down the messenger RNA until a certain portion of it reaches, the initiation code. In fact, what I'm going to do here, just for consistency's sake, I'm going to move the small subunit up here. Well, no, I will. Okay, no, I actually will move it. Okay, small subunit. All right, so now we got the small subunits up here. It's walked down the RNA until it's reached a specific point. And a certain part of that small subunit is now over the initiation codon. Now the next thing that happens is a tRNA that has the UAC anticodon, what we call the initiation tRNA, is going to make a loose attachment. And I'm going to represent the tRNAs kind of like this, okay? Just make it nice and simple. Here's our initiation tRNA. That tRNA carries the amino acid methionine, represented as MET. If you're a prokaryote, it carries a modified methionine, the informal methionine. But most things. So, it binds here. Now, we've got this assembly, and now the last but not least, and we can get rid of that cap binding protein at this point. It's done its job, away she goes. Now, the final thing is we're going to have the large subunit bind to this whole assembly, and we're ready to roll. Now, this complex is what we call the pre-initiation complex. There are other initiation factors, by the way, that play roles in this thing too. But anyway, we assemble this baby together. We're ready to start the process of protein synthesis. Now, it turns out that sequences in the untranslated regions in poly-A tail can bind proteins that can, in fact, a lot of times what will happen is it twists over and it binds proteins and it prevents one or more steps of this taking place. Like it prevents a large subunit binding, or it blocks the binding of the initiation tRNA, or any of a number of kind of things. So we can have proteins that block translation attached to these untranslated regions in the poly-A tail. We also can attach proteins that will actually promote the process. For instance, they may recruit initiation factors and help them to come in and do their job and stuff like that. So we can either promote or inhibit initiation by stuff binding to these untranslated regions. And that seems to be the major point where we're going to get regulation of translation, is at the step of initiation. Do you promote it, do you inhibit it, or what? Okay, so we've got this thing together. Now I'm going to change this diagram a little bit, and this is pretty much the end of initiation. I'm going to make a few changes in this diagram. We'll still make a really small subunit. Alright. Here's our large subunit. Now it turns out that there are three kind of indentations in the ribosome that are going to act as effectively workspaces. These three regions are one codon apart, and each one of them can bind a tRNA. And I'm going to represent them, and here I'm going to have an RNA sequence. Here we're going to have whatever. Here we have our AUG, our initiation codon. Let's say our next one is UUU, and then this one is going to be CCC, and we continue on. So this is our messenger RNA over here. Okay, now we have these 3 workbenches. And they're part of the large subunit. Each one of these are spaced 1 codon apart in terms of where the RNA is, and each one of them binds, can hold and stabilize a single tRNA. So here's the second one, and here's our last but not least one. This site is called the E, or exit site. At this point, we don't have a tRNA attached to it. And what's going to happen is the tRNA there is soon to be kicked off this complex entirely. That's why it's called exit. Okay. This site here is called the P, or peptidyl site. That's where we're going to have the ever-growing chain of amino acids. In this case, at this point, we have our initiation tRNA carrying methionine. This one over here is called the A, or aminoacyl site. This is where new amino acids are brought in, carried in by the tRNAs. So here we have at the very beginnings of elongation. Now once again, there are numerous elongation factors that are going to play roles in this that we're not going to mention. We're just going to look at the major characters, but there are numerous elongation factors that are essential for this process. So now we're going to start elongation. I'm going to try to write things down here while I'm diagramming. Our initial condition, we have a tRNA, the initiation tRNA, at the P site. We have an empty A site, that's the second codon here. So, A site empty. Okay, now we go on to step 2. A new tRNA is going to bind to the second codon, and that happens to be over the A site. So that one has to have the anticodon AAA, and that one always carries the amino acid phenylalanine. Okay, so that's step 2. Okay, now we've got the 2 tRNAs here, each carrying an amino acid. Now, the third step is what we sometimes call peptidyl transferase. Now, what that means is transferring a chain of amino acids from one location to another. And we'll see this. I'm going to show it. I'm going to write it down. Okay, what we're going to do here, and this is a complex thing, and this is what's catalyzed by that 28S or 23S if you happen to be a bacteria, ribosomal RNA. What we do here is first, we do a hydrolysis reaction here. We're going to tear this amino acid off, but we're not going to, that's a hydrolysis reaction. It does not take energy. Then we're going to somehow hold on to it, carry it over to this, the one at the A site, and now form a peptide bond. So hydrolyze amino acid from P site, move to A site. I'll put AA, amino acid, to A site. Form peptide bond. In other words, covalently join them together. Now, forming peptide bonds, of course, that's an endergonic process, and some GTP hydrolyzing initiation, or rather elongation factors play important roles in that. Apparently, at least they play a role in providing the energy for this reaction to occur. So now we've shifted the methionine from the P site to the A site. We form that peptide bond. So now we have this little short amino acid chain at the A site. Now our last step is what's called translocation. We're going to move things down. Now, what we do here, and this is interesting how this would, I'd love to see how this actually works. Okay, the whole ribosome. The entire ribosome moves one codon down the messenger RNA. Now, this is going to take me a minute to draw this. Now, the cool thing about this, the ribosome moves down the messenger RNA, but we are not going to actually dislodge the tRNAs from their appropriate codons. We will let the ribosome will release them temporarily to still stick to the codons, take a step down, and then attach them again. And what's going to happen here is this. I have to move this whole shebang now. Okay, I'm crawling the whole ribosome down. This takes more time than it actually occurs in real life. All right, so we shifted everything down. Now the sites have kind of changed. Okay. We still have our three sites, the E site. The E site is now over the initiation codon. And that still has the initiation tRNA, that's the UAC. However, don't forget, that tRNA is now empty because we removed the methionine from it in step 3. Now, the tRNA that has the 2 amino acids, it used to be at the A site, but we shifted everything one codon down. So now it's at the P site. And it has the phenylalanine and the methionine. And this is the P site. And now the A site is empty, it's over the 3rd codon. It can have a tRNA with the UUU anticodon, whatever amino acid that carries, I don't know offhand. But now we can have a new tRNA bind here. Okay, so the A site is currently empty. In effect, we are back to step 1 again. This is a 4-step cycle. tRNA with amino acids, with the peptide, the amino acid chain at P site. A site empty, that's step 1. Step 2, new tRNA carrying a new amino acid goes to the A site. Step 3, we remove these 2 amino acids from here and then attach them to amino acid over at the A site. Step 4, we shift the whole ribosome another codon down. And we continue that process along. And as it goes on, the amino acid chain grows and gets bigger and bigger and bigger. Now, it takes several minutes to describe this process here. In real life, in eukaryotes, this happens about once per second. When you're making and synthesizing a protein in a eukaryotic cell, it adds about 1 amino acid per second. Now, prokaryotes do it about 10 times faster. On the other hand, the prokaryotic ribosome is a little simpler and a lot less avenues for regulation, so you just bang the stuff out real fast. Here, we'll take a slower protein synthesis in order to have more opportunities to regulate this process. Okay, so we repeat this elongation cycle. By the way, this makes it great and consider it a probable test question. I often do that, elongation cycle, or describe the process of initiation or what have you. So now we keep on doing this. As this amino acid chain grows, it turns out the large subunit of the ribosome has kind of a tunnel beneath the P site that goes through the large subunit, so the amino acid chain actually goes down into that tunnel. And then eventually part of it will poke out of the ribosome, which has to be a pretty big amino acid chain. And when it's released, it slips through the tunnel and out the ribosome. Okay, well, that's what we got with elongation. And numerous elongation factors play a role on it. Now we're going to do the last thing, the last part of our play here. Okay, we have a really long amino acid chain. I'll just go A, you know. Okay, now, we go to our last act, termination. What we get for termination? We have a stop codon over the A site. So let's put a stop codon here. How about UGA? Did I get that right? I keep on screwing up this stuff. Is UGA stop codon? Okay, all right. Okay. So we have a stop codon here. It's over the A site. Now, what's going to happen is this. It turns out there are no tRNAs that recognize stop codons. There aren't any tRNAs out there with a ACU anticodon. Instead, what's going to bind to that stop codon is a protein called a particular termination factor. Now, the interesting thing, this is a protein, but if you look at the three-dimensional tertiary structure of that termination factor, it looks remarkably like the tertiary structure, the three-dimensional structure of a tRNA. And a matter of fact, I did the two-dimensional diagrams, the crucified tRNA. In 3 dimensions, it folds up and you get something that's shaped roughly like kind of an L. That's what a tRNA looks like in 3 dimensions, with the amino acid over here. Okay, now, it turns out this termination factor has a tertiary structure extremely similar to that tRNA, even though it's a protein. It's not a nucleic acid, it's a protein. But that particular termination factor recognizes and binds the stop codons just like a tRNA does. In effect, we're putting an imposter into the A site. This termination factor looks like a tRNA, it smells like a tRNA, it quacks like a tRNA, but it really is not a tRNA. However, the ribosome, in a sense, doesn't realize that. So the ribosome is going to try to continue the elongation cycle. The ribosome is going to cut the amino acid chain off and try to attach it to this fake tRNA here. And lo and behold, it can't do that. You cannot attach that protein chain to this termination factor. It is not a tRNA. You cannot make a peptide bond with that. So, you've torn the peptide, the protein chain off. Now you don't have anything to attach it to, so what happens? We release the protein chain. It's tried to do another elongation cycle, and it had no place to cut, to pass the amino acid chain to. Okay, so that happens. We've let go of the amino acid chain. Now, other termination factors come in and start breaking everything else apart. Okay, so we have other termination factors come in. They get rid of this termination factor. The amino acid chain's gone. They remove the tRNAs from their workbenches. And finally, they separate the large and small subunits from each other. And finally, they remove the messenger RNA from the large subunit. And now, everybody has exited stage left. And now we have the birth of a new protein. And that's our process of translation.
Wait. Oh, no.
Oh, I'm sorry.
I didn't know what it said. Other blank.
Okay. Other termination factors remove the other components. Okay. And over here, of course, the amino acid chain gets released because it can't be attached to anything else.
Okay, other termination factors removed.
Remove component.
Remove all the other components, like get rid of the tRNAs, get rid of that other termination factor, separate the large and small subunits and push the RNA off of it. And that's termination for you. Okay, now we have that. Now, of course, in protein synthesis, one of the big things is proteins have to fold up. As they're being synthesized, you'll have parts of them dangling out of that ribosomal tunnel. I'm going to make a crude diagram here. We also have what's commonly known as polysomes. In other words, many ribosomes attach the same messenger RNA all in a row and reading them. And now, of course, you have proteins of different lengths as we continue to get. Now, when things start popping out of that ribosome tunnel, what we're going to do is attach these proteins called chaperones to it. And they help the protein to fold up properly. Without the chaperones, these things would not fold properly. Many times what would happen is we have a dangling amino acid chain here that's unfolded. It's got exposed hydrophobic regions. One over here. They swing back and forth, stick together, and you got yourself a glob instead of a functioning protein. So the chaperones are going to be essential to keep the proteins, the proteins from sticking together during translation, and also giving them an opportunity to fold up into functional proteins. What we see is apparently many times, even with all this help, that fails. If so, and you end up with a misfolded protein, it gets tagged with the death warrant, ubiquitin. Ubiquitin, of course, that's small protein covalently attached in a chain to target proteins, and that's the mark of death. That targets it for destruction of the proteasomes. So, in protein synthesis, it's fold or die. It's kind of like we used to have in Sparta, the major rival to Athens in early Greece, because Sparta was a hard scrabble area. Everybody had to contribute, so older people were expected to walk out the city gates and go out and die. Babies that did not look healthy were simply abandoned and let to die off the city gates. Protein synthesis is like that. If you can't perform a useful function, folding up properly, you're going to get tagged with ubiquitin and you die as well. So that's pretty harsh, just like Sparta. Okay.
So this what you just did there, just showing us how to shape the protein?
Yeah, All right, well that covers protein synthesis, and what we're going to do is when we come back is we are going to be looking at the process of regulating translation. We've got all major players in place.